The title of today's webinar is How to Use Genetic Insights to Drive Better Results for Your Clients and Boost Retention. This is very much sort of targeted towards health and fitness professionals. That's predominantly who we work with through Fitness Genes, which we'll touch upon a little bit later. But what we want to do today is really kind of delve into a couple of the genetic insights that we provide. I mean, we provide a whole range across multiple health and fitness categories, but we've just picked out a couple for today's webinar that we're going to just provide a bit more detail around and explain how this can be used within programming, both for working out and also for nutrition as well. As I say, we've got myself, and also, sorry, as a bit of a means of an introduction. So my name's Alex. I'm product specialist. I've been with Fitness Genes for over 10 years. So if you've ever kind of engaged with Fitness Genes either as a professional or as a consumer, you've likely received emails from me or been on our website, which I kind of maintain and that type of thing. So a whole range of activities, including working closely with Stuart to produce this content as well. But if you're unfamiliar with Fitness Genes, just as a means of a very sort of quick introduction before we get into the content. So we're a health and fitness DNA testing company. So we provide consumer reports, but we also support health and fitness professionals. So we've got over 12 years experience in researching, creating, and also delivering client DNA reports. So founded in 2013 by Dr. Grice and a handful of others. We've been in this space for over a decade now. In that time, we've built up a library of genetic reports. So we very much see ourselves as a research company, essentially. So we go and look at peer-reviewed research associated with genetic variants and their influence on health and fitness and exercise, and we produce reports for our clients and also the professionals we work with to actually sort of follow recommendations from. So a library of over 187 reports to date, and I say to date because this is an ongoing project for us at Fitness Genes. We're constantly looking at new research and developing new reports all the time. So 15 health and fitness categories. So looking at heart health, brain health, muscle building, weight loss, inflammation, women's health. We recently released a methylation category as well back in May. This is, as well as sort of having that wealth of experience, we're also kind of always looking towards the future and also adapting our reporting and our products as new research becomes available. As I say, we support health and fitness professionals. So we launched our professional service back at the start of 2024. And recently we've become a SimSpa approved education partner. So, we identified the need through the professional work that we do to actually help health and fitness professionals to better understand genetics and how to use genetics within their programming. So we've created educational content, which I'll also touch upon later, but that has also been sort of verified by SimSpa, and we now have our first education course that's been recognised by them as well. So that's Fitness Genes. We also, as I say, we've got Dr. Stuart Grice sat waiting very patiently as well. So just as a quick intro, so Stuart or Dr. Grice is one of the co-founders at Fitness Genes and also is our Chief Science Officer. So very much sort of directs our scientific vision and oversees the development of the reports and the product generally. He's a former postdoctoral researcher at the University of Oxford and was situated within the functional genomics department and is also a published author and academic tutor. So Stuart, first of all, welcome, and also please let me know if I've missed anything on your own personal introduction there as well.
No, the introduction's great. The photo, not so good. I think we need to improve that one.
I'll try that. We can have a photo or a team headshot day at some point. For now, I think that works well. Welcome and thank you for your time today as well.
No problem.
Great. I think this is mainly going to be led by Stuart. He's very much the one who has the better understanding of genomics, given his background and also the founding of the company. So I guess, Stu, as a starting point, would you mind just kind of giving us an intro into kind of, the science of Fitness Genes and why we see DNA testing as being so critical with understanding an individual's health and fitness?
Sure, yeah. And I think this is a good slide to start on because be it personal trainers, clinicians, people just wanting to better understand their own health, the one thing they all have to deal with is a wealth of diagnostics, ideas, products, services that generally are quite overwhelming. But everyone really needs to distill it to the personalisation level, and this is really why a lot of these facets are key, be it DNA testing, blood testing, microbiome testing. And I guess the core to it is understanding your biology via traits. So if we can pop to the next slide. So really, when you're looking at any of the analysis on the diagnostics, be it genetics, be it bloodies, you're trying to understand particular facets of human physiology or behaviour, and as I will call them, this biological traits. And they ultimately are combined of two things, really. Is your genetics, and what is then the environmental component. When I say environmental, that can be lifestyle. We can understand how the environment has influenced us by doing blood tests, for example, by taking epigenetic tests, even though they're a new concept. Microbiome tests, for example, we really need to understand both our genetics and our environmental sort of setups to really understand our biology, which then we can use that understanding to make better health and life choices for ourselves. So here's just a sort of an example of a person, lots of different traits here: biological sex, female, hair color and eye color. These are genetic traits. They're highly genetic. There's small amounts of environmental impact that you can have to change those. Then height, body mass index, bench press. These are risk of type 2 diabetes. These are traits that have a combination of environmental and genetic risk or association or contribution. And then you get towards more complex, incredibly complex traits like behavior. If you're introverted and extrovert, there are genetic components to that, but of course there's lots of environmental interactions, how you're brought up. And then something like spoken language, English, it's fully environmental. There's no genetic component to which language you speak. So really what we've got here is an understanding if you've got all these different biological traits, some have a large genetic component, some have a large environmental component, and many are combinations of the two. So really, to understand and start to think about precision healthcare, or understanding how we personalise a training program, a nutrition program, understand someone's disease risk, we need to understand both of these facets. And even though the zeitgeist moves around in the industry with, for example, epigenetics, microbiome, for me, the core understanding components are your genetics primarily, and then how they manifest or are translated through an understanding of someone's environment. So I think we've got two examples, haven't we, Alex, if I remember correctly? So I'm going to look at two examples of genetic variants to communicate how genetics and why genetics is important for enabling us to make decisions, personal decisions or decisions for your clients. The first one is associated with personal training. It's quite a famous one in sports science. It's the alpha-actinin 3 gene associated with a protein that's in muscle. And the second one is a apolipoprotein gene that's associated with increased appetite and obesity, specifically when you take or eat saturated fats. Now, this is quite interesting because this is a genetic variant that only manifests its effect in particular environments, and it does so via changing the epigenetic markings around the gene. So I think this is a very nice one to actually show that epigenetics and genetics, or the environment and genetics, are not separate things. They interact, and understanding of that interaction is really important for making health decisions.
Okay, so just to take it back a second, so I guess our kind of perspective on personalised health is that genetics is like a single piece of quite a complex puzzle, and also has varying degrees of influence depending on the complexity of given traits and how the genetic component interacts with other components, including lifestyle and environment as well.
Yeah, so I again, my background is in disease biology as well, but I would say something like Alzheimer's, highly genetic. Breast cancer, highly genetic. Diabetes, actually, surprisingly, I think it's 60% genetic when you look at the genetic contribution. Then go to something like lung cancer. That's very, strongly environmental. And of course, it's because of a lot of the data comes from smoking. Now, again, I'm digressing already, but I wonder if you looked at the incidence of lung cancer now versus in 30 years' time when smoking is removed, you might find it becomes more genetic because you remove that big environmental exposure, i.e. smoking. So saying something's genetic or not genetic, it very much depends on the population and who you're looking at. But ultimately, yes, spot on. I will be quiet now because I'm going off piste.
Great. No. Let's get started. So the first one that you're introducing is actin 3.
Yes. So this is genetics 101 in one slide, but we have chromosomes that contain DNA. This DNA contains genes. We have roughly 20,000 protein coding genes. The way the gene operates is you got your the trigger fingers going again. The way the gene is expressed is via an intermediary molecule called messenger RNA, not really important for this call, but ultimately the genes will then express proteins. And these proteins are the signaling molecules, your hormones, your receptors in your brain, some neurotransmitters that actually do and drive all the physiological actions in your body. So we're looking at alpha-actinin 3 protein. So pop on to the next one, Alex. That's okay. So alpha-actinin 3 is a component of skeletal muscle, specifically. You find it in your muscle. And it sits on the Z disc. So you've got these sarcomeres, which enable your muscles to contract, how you can pick things up and they get bigger and you add more protein into them as you get stronger. But this particular protein has a few specific functions. First, it's a structural support. So it stabilizes the filaments in your muscle that enables you to pick up mechanical tension. It allows for explosive power. So it enhances, because of this structural component, the ability to have rapid and forceful contractions. It also helps with damage resistance. So what I mean for this, it actually mitigates things like DOMS, which is very good because it helps support the muscle and then creates the signaling processes that help drive muscle recovery. But this is actually in energetic terms at expense of endurance. So there's a little trade-off from having this ability to create explosive power, which kind of makes sense. You know, there is endurance versus power trade-offs in biology. And of course, because of these properties, it influences fiber composition. It's involved more in the promotion of fast-twitch muscle fibers. Again, apologise for the 101 on molecular biology, but here's a little snippet of the gene. So you've got here the ATGCs, which I'm sure Jurassic Park has taught you all about, if not more of your learnings. And what I've done here is just split them up into what we call codons. They're sets of 3 bases. So these 3 bases are what tell the machinery in the cell which amino acids you add in a string to create the protein that will then become alpha-actinin 3. So just pop on to the next one for me, Alex. So here we go. So you've got the kind of your 3 codons. It's translated into messenger RNA, and those 3 codons code for aspartic acid, arginine, and glutamic acid, and then you get your alpha-actinin protein. So, in a nutshell, the DNA is a code. This code can be read by proteins in the cell. These proteins, of course, are expressed through the genome, and then these derive which amino acids are in the protein. So in this case, in this little section, 556, 557, 558 are those 3 ones. Sorry, yeah, you can, it's probably best you carry on. Cool. So obviously we just explained that, but 30 to 60,000 years ago, again, with evolutional history predictions, it's always a bit off, but generally we think this is where it happens. There's a base substitution that occurred. So instead of having your C and corresponding G in this region of the alpha-actinin gene, so the actin 3 gene, we now went to a T and A. So what does this mean for the protein? So this change in base also changes a base in the codon. So what we have is a shift in the code. So we've got UGA here just as a point in messenger RNA. Thymine, they use a uracil molecule. I won't go into the details of why, but ultimately what this has done, because instead it's a UGA now, it actually doesn't code for the amino acid it should code for. It actually codes for what is called a stop codon. So some codons go, we no longer need any more amino acids on this piece of on the amino acid chain on the polypeptide. We will stop translation, and that will stop and truncate the protein. However, this mutation means that actually instead of getting the full wild type protein, as we call it, the full length wild type protein, we get a truncated protein. So people with this genetic change will not produce full length alpha-actinin, they'll produce a truncated protein. Okay, so this is just a bit of a review of that. So we've got kind of 2 alleles here, as we call them. You've got the R allele, and that's just because R is short for the arginine amino acid, and X is because it's the stop codon. And the R produces full length, the X produces the truncated version. Okay, so again, a bit of sort of basic genetics here. We obviously have two versions of each gene. Two versions of a gene, one from our mother, one from our father, because of the two sets of chromosomes. So there are three combinations of genotype from combinations of alleles that we can have: RR, RX, and XX. So RR, both producing full length. RX, one of the alleles is producing full length, which is good enough. And XX has no alpha-actinin protein produced. So these people have alpha-actinin. They have no alpha-actinin 3 protein in the cells, in their muscles, and we've just got a little bit of statistics here from our membership population. So this is, I think, on about 50, 60,000 people, and you can see about 21% of our population do not produce this protein at all. So just segregate that into two. So what does this mean? So on the one side, you've got the RR and the RXs. So you've got your protein localized at the Z disc. So that functions what we'd consider or call in the wild type sense in the muscle. But on the other side, which is the XX genotype, the lack of protein there leads to Z disc changes in the Z disc properties, which leads to changes in the muscle. Now, this is not pathogenic in a sense, although actually people who are XX are more likely to get sarcopenia. And actually, when people have particular muscular degenerative disorders, actually it means they present earlier and more severe. Generally, people can be very happy with XX. It's not life-limiting in any way. It just changes the properties of the muscle. So, Alex, sorry.
Well, yeah, that was going to be my question, actually, because for myself, I actually have the XX genotype. So you were saying that there's no kind of like health risk associated with that. It's more looking at muscle performance.
Exactly. Again, because should we pop to the next slide? Actually, I think it's on there. So this is the three genotypes again, sort of set out, and what you've got here is a list of phenotypes or changes in physiology that are associated with the different genotypes. So these are all from the literature. So you've got fast twitch muscle fiber diameter, response to resistance training, baseline muscle strength, post-exercise muscle damage, movement range, so stiffness and movement of the muscles, and then aerobic function and performance specifically. So as you can see here, when you've got alpha-actinin protein, you're more likely to be stronger, have higher baseline strength, and have less muscle damage. Now you can see why this gene becomes a risk factor for sarcopenia, for example, because if you don't have it, your baseline strength, at least associated with this one locus, decreases. Now, as you also see, though, actually there's some advantage to be XX, particularly within the aerobic function. So more for endurance athletes. Is this where you drop in your marathon time now, Alex? I know you've been.
No.
Yeah. So again, I know this is the one thing we see quite often, and again, maybe for another conversation, but there are many trade-offs in biology, and you see very much about the anti-aging movement where people are obsessed with about optimization. Actually, in biology, you very often find trade-offs. So here, where you've got a benefit for aerobic performance and movement range, which you can think in terms of longevity and health, it's quite an important thing, particularly with movement ranges and VO2 max. We know that this can be impacting your ability to increase VO2 max, or at least cardiovascular functional performance. Actually, the downside is you have a slight reduction in your ability to maintain muscle mass, or at least you have a lower baseline. So here's a little trade-off example. But of course, when you see this information, I appreciate this is one variant out of the many millions, but you can start to think about how you would then target your personal or your client's response to it. So I've just picked one very small example here, which is around, on the next slide, sorry, Alex, which is around kind of post-exercise muscle damage. So alpha-actinin 3 is associated with increased exercise damage, and within our traits we look at that, and you can start to think about particular supplements or how you stagger your rest between workouts. You can start to think about how you target recommendations based on this information.
So I'm aware that we've covered quite a lot of ground there, from sort of like all the way from sort of genetics 101 through to providing this example for actinin 3. So just in a nutshell, what we're saying is that changes that occur within the sort of base DNA code have a resulting effect on the types of proteins that an individual produces. And it's through that understanding of an individual's sort of unique protein production, or sort of in this case, the lack of proteins produced, that we can actually start making recommendations around training, nutrition, supplementation.
Yeah, in a nutshell. And of course, this is one variant in very specific one. Of course, there's lots of interaction between these, but that's absolutely correct. And this is an exact. Sorry, are you going to go and?
Well, because this is, I guess, the point that we were going to draw upon, and how that can then be used. Just thinking specifically about kind of our audience today within sort of programming for a client. So I know Actin-3 is one that we look at for a whole range of traits, including post-exercise recovery rate, recommended training volume, rest between sets.
Mm-hmm. Exactly. So if you think, again, you're asking the neuroscientist biologist about physiology here. But so if you think about lifting weights and hypertrophy particularly, you've got the two components. You've got mechanical tension, you've got metabolic stress. So metabolic stress is more about the energetics. So alpha-actinin-3, we've shown, can increase your ability to maintain mechanical tension, but actually maybe not as beneficial for maintaining or at least dealing with metabolic stress. Now, if you think, but if you think about the biology that underpins those two components, so you've got the fibre type, which again, the energetics profiles are going to be different. So people will have genetics that steer people to have ratios of one versus the other. So actually the makeup of the muscle will be different. That will then obviously impact how easy it is for them to deal with mechanical stress as well as mechanical tension, as well as metabolic stress. You've got lactate clearance and threshold. So obviously lactate, which was actually the maligned molecule, but actually it's just a byproduct which then is used for energy, and it's actually the hydrogen ions that cause acidosis. So we know there's mutations in the transporters of both lactate and these hydrogen ions. So some people can shuttle it back into the muscle, so therefore they can use it, so which means it extends their endurance profile. However, the people, so actually that's very beneficial for endurance exercise because you're not having the lactate burn whilst you're using lactate as an energy source. However, when you look at the other side of that, actually for those people who have the poor transport, actually they have increased metabolic stress and actually have increased hypertrophy response. So if you think of someone could deal with a high mechanical load, mechanical tension, and has an increased profile of mechanical stress, actually a higher volume, so higher weights, lower reps can get their time under tension than people who have muscles that deal with the metabolic components better, but actually will fail, or you'll have more likely to need a longer recovery period if you lift heavy weights, i.e. the x-axis, for example. But again, there's lots of other genes in ACE and VEGF factors and other genes that are associated with how you build a polygenetic risk score to say someone scores highly on strength versus endurance fiber type, someone has good lactate clearance and threshold versus bad lactate clearance. And then of course there's the inflammatory properties as well. Some people have a higher inflammatory response, which is good for hypertrophy to a point. So you start to build these different facets of biology, and then you can go, okay, well, I think you're going to respond more to higher weight, lower rep because of these parameters, or at the other end of the spectrum, you're going to respond to the higher number of reps, but actually lower the weight because actually it can impact your recovery, for example, after a workout. Does that kind of make sense?
Yeah, so Actin-3 would be one of the ones that is used within the calculation of these traits, but it wouldn't be sort of like
it's
solely
not exclusively
down
Actin-3 for sure. There's many other genetic variants that are used to calculate these parameters.
Okay, excellent. We're just going to take a little pause there, and I'm just going to check to see if anyone has questions as we're going through the content, please feel free to drop it into the chat, and we'll pick those out. We can either leave those till the end, or if they're relevant as we're going through the content, then please feel free to use that, and we'll come to those as well. So I can see Tristan from Sheffield has also joined us. So yeah, please feel free to use the chat just to let us know who you are and where you're joining from, and how warm it is in your room currently, because I'm absolutely roasting. I'm sorry, I've got some building work going on outside, so I've closed all my windows for the school, and I'm paying the price. But yeah, so that's Actin-3. I'm aware at the time we want to make sure we have enough time for APOA2, which is associated with fat metabolism. But yeah, for anyone who's got any questions, please feel free to use the live stream, and we'll come to those as and when we can. So Stu, I'm going to jump you back in.
Cool, no problem.
And we're going to look at the second gene of interest today. But first of all, if you'd like to touch on kind of genetic environment interaction as well.
Sure, yeah. So I think this is quite an interesting trait because it covers both environment, genetic interactions, but also involves epigenetics. And the one thing I find a lot is in this field is people go, well, you work on genetics, but isn't it all epigenetics? We have a lovely video that maybe you can post afterwards in the feed about actually describes our perspective on epigenetics if you want to learn more. I think it's on YouTube. But ultimately, the epigenome or the epigenetics manifests only through the genome itself. So methylation, which is probably the most commonly known one, these are associated with DNA bases in the genome that can be mutated or not mutated. Then, of course, you've got the environment that drives changes in the epigenome, but that only manifests through proteins, and the proteins are encoded by the genome. So when people say it's only epigenetics, no, these are highly, tightly interlinked processes that require each other to manifest the traits that we know, love and study in biology. And then just a little technical term when we talk about epigenetics, because I notice some people use epigenetics and environment sort of interchangeably, but actually epigenetics is a study of how the cell regulates gene expression without altering the DNA sequence. So what we've seen with alpha-actin 3, there's a mutation in the DNA sequence which stops the gene being produced. Epigenetics just adds chemical moieties to the DNA or to histones or through other factors to change the way a gene is expressed. And if you think about a lot of mutations that stop proteins being produced are generally quite deleterious and linked to disease. That's why alpha-actin is quite an interesting case. A lot of the mutations we see just alter expression levels or change the functionality of the protein in some way. But again, I think the take home from this slide is genetics, epigenetics, and environmental are interlinked, and you cannot ignore any facet of them. However, genetics is the master, epigenetics is the servant.
And I think although we're a DNA testing company, I think we all see environment as being as important because that's the part which you can manage as well, right? So that's where kind of like the value can be found in terms of making alterations to your lifestyle, to your exercise, to your nutrition, to have an effect on your total health and fitness.
Sure, and I think this is why this trait's interesting because it shows an environmental genetic interaction. Should we get into it? Okay,
yes.
just on that point. So an environment genetic interaction is where in one environment the risk allele has no effect or no differential effects from what is the non-risk allele. So here we've got environment A. It's lovely. It's a very academic slide. We've got the AA and AG, which confers no risk, and you've got GG, which is considered to be the risk, but the outcomes are the same, and that could be anything from risk of something, how you respond to something. Now, if you're in a different environment, as you can see here, the GG now has a different outcome. So it's the context of both the genetic variant and the environment that lead to a particular outcome. And this is where I think a lot of people get confused when they go, obesity is not genetic because look at the post-war years and everyone was thin, etc., etc. But actually, it's the combination of the environment and the genetics that leads some people to be a lot more susceptible to putting on weight than others. And we've got an example here. So we're looking at apolipoprotein A2. So apolipoproteins sit on sort of high-density lipoproteins, low-density lipoproteins, moieties with fat molecules in. Basically, they're associated with the absorbance of fats, the transport of fats around the body, and cholesterol and other lipids. So they're really important in the body for responding to dietary intake of fats. Now, there's a genetic variant outside in the regulatory region of this ApoA2 gene, which codes for apolipoprotein A2, that only increases someone's appetite and then also the added risk of weight gain if you're eating over 22 grams of saturated fat a day. So this is very interesting. So why is this? How is this happening?
Sorry, Stu, I think you just dropped for a second. Was that over 22 grams of fat per day, saturated?
Sorry, yes. So if you're eating over 22 grams of fat per day, the genetic variant confers increased appetite and increased weight gain. Did you? catch that one?
Okay. Yeah. No.
Good.
Thanks for clarifying.
No problem. So this is so as we described with alpha-actinin 3, there's a mutation that changed an amino acid, actually converted it to a stop codon, led to the truncation of the protein. The protein was useless; it would be degraded in the cell, and therefore the muscle could not have that protein, and it could not elicit the positive hypertrophy, protective muscle building effects in that person. So therefore, you have to design programs and nutritional strategies around that. Here, we've actually got a mutation that's not in the gene. So there's just to explain this, the grey bit is what is called non-coding. The non-coding bits by genes are generally involved in gene expression regulation. The P is just a promoter where you will start to produce the messenger RNA from the little coloured sort of skittly things there, which are the basis of the DNA. So we've got this change, a T to a C change in the non-coding region, which we know is associated with gene expression regulation. So on to the next one. So interestingly, we've just talked about a T to a C change. So when we have a T, you generally get an increase in, or you can be eating saturated fats over 22 grams, and you still got high expression of this protein. This protein, of course, is involved in transport of many lipids, and as you can imagine, it's very important when you eat fats or with cholesterol as well. So saturated fat often comes with cholesterol. You need ways of increasing the amount of proteins associated with the absorbance and the transport so you can utilize those nutrients very well. Now, interestingly, for the people who have the C change, the gene is not expressed, and actually the gene is expressed a lot lower, specifically when you eat over 22 grams of fat per day. Now, why is this? So we talked about epigenetics. One of the ways you reduce gene expression or lower gene expression is by methylating the regulatory regions of the gene. And these methylation markers are attached to one nucleotide specifically, and that's a cytosine. So the presence of a cytosine increases the chances of methylation in the regulatory region of this gene, which then lowers the expression of this protein. So what you've got here is an environmental change or an environmental situation, lots of fat, a genetic change, and then a corresponding epigenetic change, which leads to a differential expression of the gene. So it is actually the genetic mutation that has caused the epigenetic change, not directly the consumption of fat itself. It's a combination of them. So how does this elicit any effect on the person? So pop to the next one. A little bit complicated, but well, just the sort of the high level here. So we know that sort of apolipoprotein regulates the absorption and transport of fats. Obviously, the brain and the body needs to know what food we've had, so therefore when you have these different changes or fluctuations in foods, you'll get different interactions with brain chemistry to basically increase or decrease appetite accordingly. However, when you have this erroneous reduction in ApoA2 expression and therefore a reduction in the apolipoprotein A1, you get misregulation of neuroactive metabolites, kynurenes, and particularly around tryptophan metabolism, which is associated then with serotonin production in the brain. Now, serotonin is a very important repressor of appetite, promotes satiety and food intake. So what you have here is the reduction of this protein leads to a change in neuroactive metabolites in the brain, which leads to an increase in appetite, which then leads to overeating. And this is why ApoA2, this variant of ApoA2, is linked to a risk of obesity. Now, on to the next one. So, biology is complex, as we've of course. So what I'm doing here is just trying to show you how we've been talking about single genes here, but now actually the reality of what we do at Fitness Genes and what we build at Fitness Genes is what you call polygenetic scores or polygenic risk scores, where actually it's multiple hits, multiple risk hits or multiple positive hits. For example, you could have lots of positive variants associated with power or endurance, but also you can have lots of hits associated with a risk of diabetes or a risk of overeating. And this is a very simple example of how some of these genetic variants interact synergistically to produce increased risk. So I've just added an extra genetic variant here, FTO variant. It's reasonably well known. So the basic mutations in FTO increase appetite, increase people's food desire, so they're more likely to overeat. So we've got person 1 here. They have no risk in FTO. They have the risk alleles for ApoA2, so the ones which has been associated with increased eating. However, they are not eating over the 22 grams of saturated fat per day. So in the context of this person's environment and genetics, they've got average appetite, and their weight gain risk we'll consider average. Now let's look at person 2. Person 2 has the risk for alleles for FTO. This increases their appetite, as we suggested. This actually then leads to them to increase their intake of saturated fat over 22 grams. However, they don't have the risk factor for ApoA2, so they don't have that additional increase in appetite. But still, this individual with this one risk variance, has an increase in appetite and an increase in weight gain. Now let's look at person 3. This person has the risk alleles for FTO, the risk genotype. This will lead them to overeat. This can include both sugars and saturated fat, which can, in certain circumstances, increase them to over 22 grams of fat per day, just because they're pursuing fat because of their genetics. Because they're over 22 grams of fat per day, we have the modification of the ApoA2 locus, because they are also carrying the risk variance for ApoA2. We know this then creates an increase in appetite, which then can lead to an increase in fat intake and a large increase in weight gain. So this is just a very simple way of showing how genetic variance can interact to increase risk. And it's really important when you start to build out. As we said, we've got lots of different traits associated with different components of weight loss, in muscle build, in endurance, in cardiovascular risk. We start to think about all the genetic variance in context. So do you want to pop to the next slide? So again, just thinking about the risk associated with saturated fat, of course, from a translational point of view, replacing meat for vegetarian protein sources will reduce we reduce saturated fat. So adding 5-hydroxytryptophan, which is associated with an increase and moderation of serotonin production, which has been shown to increase satiety and reduce calorie intake, obviously via repressing appetite. And then, of course, the tracking as well, actually knowing how much saturated fat you're having in your diet. So these are ways of intervening with some of these genetic variants. So you have people think about obesity be kind of a generic broad term, but here, specifically saturated fat can drive this person's drive their appetite. Now, this is not saying saturated fat shouldn't be eaten. It's an important nutrient. However, understanding that saturated fat can be a driver for, I think it's 10% of our population have this ApoA2 risk genotype. It can confer an increase in appetite and actually be sabotaging their diet. So we've had people who've gone more ketogenic and said they very much failed on the ketogenic diet. They were always pursuing food and actually they were pursuing carbs. And this could be one of the reasons why that was happening, because you're increasing your appetite because of this interaction with saturated fat and ApoA2. And Alex.
Yeah, just a quick question. I guess, how easy do you think it would be to unknowingly consume more than 22 grams of saturated fat per day? What might that typically look like if someone wasn't following the recommendation?
It's not super easy unless you start, if you've got a diet which is ketogenic, for example, it's very easy, very, very easy. So it depends. A high vegetarian diet, it's difficult. So again, there's the environmental context, a very good point, where if you just happen to be a vegetarian, which is independent of that those genes, you will not see the risk conferring through this locus, because just by chance you're vegetarian. Now, if you just happen to have a high meat diet, and I'm not getting on the carnivore herbivore debate here, but you can just see how what would be a good diet to one person is not necessarily a good diet to another person, and the environment we've described here, which culturally someone can be a vegan and culturally someone can be a carnivore, but actually it's the genetics that ultimately will manifest whether that is positive or negative, if that makes sense.
Yeah, no, absolutely. And also your point about, these are two genes, but I know we've spoke about, previously looking at things like beta oxidation and actually accessing fat as a fuel source. So if then people have variants associated with reduced ability to access those fuel sources, and that's another risk factor combined with FTO and APOA2. I guess that kind of adds another layer to this again as well.
Exactly, and what you're describing here is a complex polygenetic score, and this is gone. And just to doff the cap to the training program, we talk about how we build polygenetic scores, specifically in that case for VO2 max trainability. How you get, it's kind of like a multi-hit model, where the more variance you have pointing in one direction or the other can actually make fat, saturated fat or fat in general, a very positive food source for someone in terms of obesity, or very negative food source in terms of putting on weight. So absolutely. So people say carb diet works, high carb, low carb, high fat. Well, depends. Very much depends. And again, so this is just linking into that. So here we've got like 3 health goals here. So muscle hypertrophy, endurance response, and fat loss. And as you can see, they all consist of very different components. So appetite, impulsive eating, compulsive eating, metabolic efficiency, responses to macronutrients. They're all different things that actually can lead you to overeat or undereat. And actually the way you would target nutritional strategies, if we discussed with, vegan versus carnivore and carb versus fat in the diet, will change depending on the portfolio of traits you have. Again, with the training, so for this individual, I'd just like to point out there are more traits associated with these health goals. This is just for sort of visual purposes. For this person, would require more help with their hypertrophy, particularly with recovery, but this person appears to have better endurance profiles. So again, you can see how all these traits create a mosaic of positive or negatives, or things to work on, things to be happy about, or risks and low risks. And this can enable you then to start to navigate where the quick wins are for changing your diet or your training program, or helping someone who comes to you, a client that comes to you asking for help. It gives you, along with the environmental data, just as a point actually in terms of what we do at Fitness Genes. We are a genetics company, but part of our platform and our patent, actually we're the first company in the world to actually have a patent that actually uses genetic and non-genetic data to actually come up with results. So we actually have non-genetic data that actually drives some of the results within the system as well. So really it gives you a platform to sort of know where to target and how to start focusing on the particular risks or opportunities on someone's health.
And I guess sort of looking at this, that's kind of the point, is identifying the individual components and where the risk lies, so that you can kind of prioritize different aspects of a health goal for the individual.
Exactly, yeah. Okay. And of course, because environmental data, because people say, well, genetics is genetics and it's stabilized. Well, that's true, but actually, because we've seen these genetic environmental interactions, actually as your life changes, if your diet totally changes, or if you go through the menopause, or if you get older, actually the context of how your genetics manifests positive or negative benefits actually changes. I think we talked about in one podcast IGF-1. High IGF-1 is very important for growth, not so good for longevity and chronic disease. So again, how you would strategize different nutritional or exercise regimes would depend very much on, in that case, age.
And those recommendations and strategies are provided through Fitness Genes as well.
Yes.
Good. Always.
But you knew that already.
Yes. Yeah. That was a very leading question.
Yeah.
No. Thanks to you. I mean, I'm wary of the time, and I want to make sure we've got a bit of time for any questions people have as well. So, I'm aware we've packed quite a lot in there, looking at Actin-3 and APOA2, but hopefully those prove, quite useful examples of, just how we're able to use the genetic and lifestyle data to try and, I guess, segment based on risk factors and provide those personalised recommendations. As I said at the top of the call, and as Stuart alluded to a couple of times during that, we do offer professional training for health and fitness professionals. So we're a SimSpire education partner. We launched our first education course earlier this year called The Genetics of Physical Performance and Health. These two, the Actin-3 and the APOA2, are 2 example genes that we actually explore through that education course. So 4 modules, it probably takes around 6 hours to do in its entirety, so it's very sort of thorough. where we look at foundations of genetics, genetic variance and performance, polygenic traits, as Stuart mentioned a second ago, and interaction, and then applied genomics in fitness and health. So actually, how do you take this genetic data and use it within programming for clients within a health and fitness context? So this is, and yeah, Stuart, you were heavily involved in the production of this course alongside the rest of the education team. So this is something that has been produced in-house by Fitness Genes.
Cool. Should we have some questions?
Sorry, before we do, the sort of my part, actually for attending this webinar or viewing the recording of this webinar, we're giving people the opportunity to actually access that course at a much reduced rate as well. So we've actually knocked 60% off the price of the course for the rest of this week. So if you're interested in actually taking this course and going through the content, some of which Stu has sort of described in this call today, you can actually go to fitnessgenes.com/pro/education and use the discount code webinar60 to save 60% on that education course. It's also included in all of our partnership agreements. So if actually you're looking to use this within your own business, this is one of kind of the resources that we provide access to all of our partners with, but it is a resource that people are able to purchase and go through in their own time as well. So as I say, if you are interested in looking a little bit more around the education and going into a bit more detail around genetics and how it's used within health and fitness, then do go to fitnessgenes.com/pro/education and use webinar60 to save 60% on all education products until Friday. That includes the bundle of the course plus your own personal DNA kit for I think less than £140, which is a real steal at the moment. So yeah, fitnessgenes.com/pro/education.
Cool. I picked up a question from Michelle.
Great. Yes, if we can.
If results conflict each other, how do you decide which one to prioritise? Great question. As I was saying, biology involves many trade-offs, and I guess it very much depends on the conflict itself. So the first thing we do is we're building in the high risk, low to low risk. So if someone's higher risk, would tackle that one first. Now, let's think of an example of a conflict. So a good conflict is, and there's thinking about endurance, would be caffeine. So we know some people with fast caffeine metabolism elicit benefits from caffeine. However, and that's through a gene that metabolizes the caffeine, which means you get the metabolites that repress the feeling of pain during exercise, and also clear out the metabolites that impact the cardiovascular system, which actually can reduce the performance. However, there's a second variant that increases the risk of anxiety and insomnia with caffeine. So as you can see here in the holistic picture, where you've got a study which goes actually, well, you benefit sort of physiologically during exercise from caffeine. However, you're more likely to get anxiety and insomnia. There is a very difficult trade-off to tease out. So for that one, I guess it really, that's where you need the conversations with the individual themselves. Personally, for me, I've just described my sort of situation. So training in the morning, I will take caffeine, but I will not be having it close to bedtime. The second one, if there's something important, like something like today, believe it or not, I can get anxious, Alex, give it up. But if there's something like really important, I would just eliminate caffeine from it because of the anxiety. So really those components of trade-offs, it really becomes quite personal. Now, I'd like to point out we are on the 187 traits, building a platform that actually ranks these conflicts now. It's something we're building out with machine learning for the last 2 years. We're very much getting close to that, so that's something we'll offer as a product in the future, so you won't have to resolve these conflicts, or at least we'll contextualize them enough for someone to understand them, as opposed to them being kind of disparate bits of information. The second point of conflict is it's something that will raise its head in biology all the time. I was on the Doctor's Kitchen podcast a few months ago now, but it's if you think about cancer versus dementia. Now, cancer is a disease of cell proliferation, of growth, as dementia is a disease where you can't replace neurons, so reduced cell proliferation. There biologically is a trade-off in its own right. And it's kind of knowing your risks there. So say, for example, if you and unsurprisingly, people with higher dementia risks seem to have lower cancer risks, and people with higher cancer risk do seem to have lower dementia risks. It's seen within particular cancers at least. So it's actually understanding your biological risks. So good example is ApoE. I would really focus on the key. If I had risk variance associated with that, I would focus that on a condition. I'd focus on that as a priority, and of course the gradings within the system can highlight that. In terms of health and fitness, and sort of the ability to respond to training, again, we've been describing genetic variants that, as I don't like to steer people towards one direction or another, but we know there are some people that have a very limited capacity for moderate to high level endurance, but maybe very good at strength, and vice versa. And I think it's just the realities of those biological trade-offs that actually need to be addressed. I hope this is, I think this is maybe gone around in circles. Does this make sense? Building on Michelle's question, I've struggled to unpick different overlapping risk factors in my diet and training. The possibility to save action has helped out. If not, I should be able to use FG to help my clients. Perhaps I need to do the course. So the course, the last component of the course, does go into how you assess these, so why you get trade-offs, why you get contradictions, and how to tease those out. So it does go through that in the latter parts of the course. That's correct.
And also on that point as well, and Tristan, maybe this is a conversation we sort of take offline, sort of through the professional service, we do have preloaded templates which kind of do highlight sort of and rank the individual report. So that's a process I've been through with the science team. So there are ways where we can actually segment the information based on your client goals. So let's have that conversation offline. A final question from Ben as well. How effective is the advice for athletes seeking multiple training objectives?
I could define multiple training objectives is my comeback there.
So for myself, I guess looking to improve on my half marathon time, but also wanting to sort of build muscle mass simultaneously. Is there a way that the information could be used to kind of achieve multiple goals at once, or should focus be placed on one given area?
Again, it's if you are so when we say athletes, if it's elite, I guess so what it can do is it can identify where your strengths and weaknesses are. So in terms of what's the best path of the trade-offs? So we described the example with alpha-actinin 3. For some people, they probably can do well. For some people, they've got a higher baseline of strength. So therefore, actually, if they're looking for the marathon time, maybe focusing on the endurance component of that is important. Now we've got other traits. So, for example, response to VO2 max. Now we know some people will not respond to the classical VO2 max training protocols. So therefore, they should focus particularly on more of the lactate threshold or the mechanical sort of processes associated with I forgot what it's called now. There's another training process anyway. So again, we can't really. There's no, and I think the marketplace likes to sell perfect solutions, but the reality is biology is complicated, and you're always going to have to deal with trade-offs. My area is more longevity than sports, but those trade-offs do exist, and it's how you navigate so with a disease, it's how you navigate the risks that you should prioritize. So as I talked about, dementia versus cancer, for example, that's what I understood from my research in my lab. Now, in terms of the trade-offs or understanding of how you should train, well, I guess you're going to, and we have it in the system, you have your endurance profiles, you'll have your traits associated with hypertrophy, particular signaling pathways, your understanding of how you can recover, and that information can enable you to navigate the best path. I hope that helps.
Yeah, no, I'm sure it does. Ben, if you've got any more follow-up questions, we're happy to kind of pick that subject up with you further as well. So I'll just give it a minute just in case anyone else does have any final questions. I'm aware we've run over slightly, but hopefully that has been interesting and has been a useful kind of hour of people's time. I'm sure it has been. Just a reminder of there is a couple of... Dickie, from your research, is it possible to extrapolate how someone is affected by genes and their response to training without necessarily having their genes looked into? E.g., if someone finds themselves falling asleep when taking caffeine, can that indicate that they might respond less to strength training? We did have this question actually at an event that we went to last week. Someone approached and said that they're confident they'd be able to identify all of their clients' traits without taking the test, and we suggested maybe they come and work for us. But Stuart, your response to that?
Yeah, no, and I think you're right. Like there's some genetic traits that are obvious: hair color, eye color. And I know that sounds a bit of a cop out, but it's true. You will personally understand your response to nutrition because you'll have particular effects. Now, again, just because I worked on neurodegeneration, ApoE, the buildup of tangles in the brain of particular proteins, of protein aggregates, that happens for 20 to 30 years before a clinical test can tell someone's got Alzheimer's. So for some traits, you don't know what's going on. Hypercholesterolemia, trait associated with increased cholesterol in the bloods. Generally, the raised cholesterol happens potentially from the age of 15 to 20. When's the first cholesterol blood test you really would do? For me, it was in my 40s. But actually, the genetic test can be telling you straight away. These are things that you are not recognizing. So, Dick, you're absolutely correct. There are certain traits that you will be able to predict, but there are other factors. Like, for example, alpha-actinin 3 and muscle recovery. I guess you probably know you take longer to recover in your friends. There's an example where you go to the gym and you just know your DOMS lasts a little bit longer than it's someone else. You can be in a running club, and we've got the VO2 max trainability prediction, and you can be doing the same VO2 max protocols as the rest of your group around a track, and you may not be seeing the improvement. So again, yeah, you're absolutely right. There are some traits that you can predict, but I'll also tell you there's some traits that where by the time you can predict it, it's a bit late.
And also, what I've found is when working with our partners, it's not always about revealing new information, but also kind of confirming or reaffirming things that they're already doing with their clients, and kind of being able to point to evidence as to why that may be something that they should or shouldn't do. So, it's helpful to be able to kind of back up kind of strategies people are already following with further reasoning to help with kind of that adherence to that particular behaviour as well. So, we always want to learn something new, but even if it's just reaffirming something, then that can be just as useful.
Yeah, and also genetics, blood data, microbiome data, these lifestyle questionnaires, they're all little sort of pieces of the prediction. It's not, none of them are covering every component of health. So actually the sort of the feelings, the perceptions, the responses, that's why we have an extensive questionnaire as part of our product, because actually we're asking these things, and it actually enables us to realize and contextualize the genetic information as well relative to someone's sort of health status and sort of eating behaviours, etc.
Great. Well, we're about to hit a full hour, so I think we're going to leave it there. One final one from Michelle. So if your genetic predisposition is to have fast twitch muscle fibers, but you don't know, is it possible through time to change the distribution to slow twitch? So this was
that's fancy.
Comment.
I like that. That's cool.
Hey, I've just learned
You've only just figured that one out.
Yeah, absolutely.
We'll learn that today. Yeah, so that you have a predisposition of leaning, but of course you can train to improve the fibre blends and fibre types for sure. But again, so if you think about the classical normal distribution, a lot of people sit within the middle. You have blends of muscle fibres. There are people that will be genetically so prone to fast twitch. They have a lot more prone to fast twitch, a lot more prone to slow twitch. But generally, there is movement. Same with body weight. We know height's a great example. Height is highly genetic, but if you're malnourished, you will never get to your height. If you are prone to fast twitch fibres, but you do marathon training constantly, you are going to move towards slow twitch. So it's not a factor of destiny. It is just a leaning. So if you're looking to, like, for hypertrophy is a great example, because your outcome is to make muscle fibers bigger, and both slow and fast twitch, the type 1s and type 2s, can be expanded, but also their properties will enable you to do the lifting in a particular way. And that's why I think it's quite interesting the way the traits are built at Fitness Genes to actually steer people in those directions. But you're spot on in terms of this. It's not a destiny. It's which door is the easiest to push open, I guess.
Nice. I think that's a very nice quote to leave it on, I believe. So yeah, if anyone has any more questions, you can email me, alex@fitnessgenes.com. Reply to any of the emails that you received in the build up to this webinar as well. Also support@fitnessgenes.com for any questions you have that you'd like to direct towards Stu. They'll get passed through there as well. So yeah, thank you very much for joining, everyone. Stuart, thank you very much for your time as well. And for anyone who is interested in looking at the education content and course further, that's fitnessgenes.com/pro/education. And you can save 60% on that until Friday, just as a thank you for coming and joining us today as well. So make sure to use webinar 60 when you are going through that checkout process. Thank you all, and we'll see you at the next one as well.
Thank you.